Hello, good morning, students. If you remember from our last lecture, we have started about the DNA damage and DNA repair system. In the last class, we discussed about the different kind of DNA modifications and damages which may arise spontaneously or that might be induced by the presence of some mutagens. So we have covered many things like this one. And then we have covered different kind of mutagens, endogenous, exogenous, physical and chemical. And then we have also discussed what kind of cells response generates after the DNA damage. So there are certain responses which are intercellular responses which we do not see. There are certain responses which are manifested by the cell at the macroscopic level that we can visualize by the eyes or by the microscope or by the certain behavior of the cells. For example, growth, population, maybe the natality, and then the mortality, the percent survival, etc., etc. We have discussed this one in detail, and then we have discussed different kind of DNA damages, mutations that may arise because of the spontaneous reactions or by the reaction by certain chemicals. We have discussed all these things in the last class in details. And here is one example how the most common base which undergoes the methylation is the cytosine C, and this one. The cytosine can undergo methylation, the cytosine can undergo deamination, and because of the methylation and after the methylation it can go the deamination and it can result into thymidine. From cytosine it can convert, cytosine can convert into thymidine, and the cytosine can convert into uracil, and the uracil can convert into thymidine. So basically that all originates from the cytosine. So if we say that because of the alkylation or methylation and deamination, a cytosine can result into thymidine or thymine as well as uracil. Similarly, if there is a uracil that can convert into thymine, and similarly the thymine can result into uracil also. If the thymine undergo demethylation, it will result into the uracil. So that can also happen. So here is one question that is in December 2018, the paper code A and the question number 50, that the deamination of bases is a common chemical event that produces a spontaneous mutation. Which one of the following bases will be formed by deamination of 5-methyl cytosine? So here is methyl cytosine. That's very important thing. Not cytosine, but it's a 5-methyl cytosine. So we know if there was a cytosine, if it undergoes deamination, it gives uracil, and if it is a methyl cytosine, it goes deamination, then it gives thymine. And similarly, if the thymine undergoes demethylation, then it will give the uracil. And here the cytosine undergoes methylation, then it is become the methyl cytosine. This will be the methylation. Similarly, the uracil undergoes methylation, then it will give the thymine. So, and if you remember in a cellular system, the methylation is greater than demethylation. Both these processes happen in the cells, but the most frequent process is the methylation rather than demethylations. So this observations gives a force to the scientists to think in a particular direction that Probably originally in the living world, it was the RNA that contained uracil, and the uracil, with the higher rate of methylation, slowly and slowly, it started converting into T, and the proportion of T started increasing, and the RNA converted into thymine containing RNA, and then RNA lose the oxygen at the 2' carbon, so the sugars of the RNA become deoxygenated, and so the thymine containing RNA converted into the DNA. So anyway, from the 5-methyl cytosine, we have already seen that it should give thymine, not uracil. Most of the people will go for the uracil, but remember, the uracil is formed from the cytosine, not from the 5-methyl cytosine, so the answer will be the thymine. Similarly, we discussed various kind of chemical agents, the acid, the bases, and then the reducing agent, then the oxidizing agent, and then the alkylating agents, and what kind of different modifications they will generate, and based on those modifications what kind of mutations will arise. And even in the modifications, generally we categorize the mutations like frame shift mutations or point substitution mutation, where one amino acid is changed into another amino acid. In the frame shift mutation, the entire open reading frames get converted into a different ORF. So all the proteins after the mutation, at the site of mutation, all the amino acids will change in a polypeptide. So that will be much more drastic mutation, and the protein will have a totally different nature and different function. So that such kind of mutations are more deleterious mutations. So anyway, we have discussed all these things also, and based on all those things we had one example we discussed last time. This one example, this question was in December 2012. This is a very representative and typical example. So where the chemical mutagens, they are tested, they are used for a mutations, and sometimes a second mutant or second mutagen is used to assess and qualify, or maybe rather that to investigate the nature of the another mutagen, to find out the mechanism of that mutagen, how that mutagen induce the mutation, in what methods, in what way. Similar example is asked in 2018. So remember, like this question can be asked again, exactly on the same pattern. So here what we see in 2018 question, that a chemist, the language is almost same. See here, a chemist synthesized 3 new chemical compounds M1, M2, and the compounds were tested for their mutagenic potential, and so on, so on. Almost the language is same here. There was M1, M2, and M3. Here is X and Y, Z. A chemist synthesizes 3 new chemical compounds in the laboratory and names them as X, Y, and Z. After analyzing mutagenic potential of all these compounds, the genetist observed that all are highly mutagenic. Exactly the same language. Some words are here and there, but it's the same language. For example, here the compounds were tested for their mutagenic potential and were found to be highly mutagenic. See, basically the words has been changed, but the end message is that these 3 chemicals are highly mutagenic. That's it, the take-home message. Similarly here also. Now again, there was a scientist, here is a geneticist. That doesn't matter. The scientist also tested the potential of mutations induced by these compounds to be reversed by other known mutagens and obtained the following results. So the scientists wanted to discover if the mutagenic effect of these mutagens can be reversed by some other mutagens. And by these experiments, the scientists can elucidate the mutagenic mechanism of that mutagen. So what we see here, these are the X, Y and Z, these are the mutagens. They produce some kind of mutations, and those mutations are tested for reversion by chemicals nitrous acid, hydroxylamine and acridine orange. So we know that about the nitrous acid, HNO2, from our previous class, that HNO2 does the transition kind of transition mutation. It does the transition mutation. HNO2 does the transition mutation. We know already. Similarly, we know acridine orange. Acridine orange is a DNA chelator or DNA intercalator, basically. Sorry, DNA intercalator. And we also know that the DNA intercalator generally induce one addition or deletion of a nucleotide that results into the frame shift mutation. So the acridine orange, similarly proflavine and ethidium bromide, they all induce frame shift mutation. Frame shift mutation. Frame shift mutation. So now when we know that, once we know that the nitrous acid does the transition mutation and acridine orange does the frame shift mutation. So basically, like dissolve, the same thing will happen here. The like mutagen reverse a like mutagen. So transition kind of a mutant which generates the transition mutation will reverse the mutation by another mutagen which also cause the transition mutations. Similarly, a frame shift mutation causing a mutagen will reverse the mutation effect by another frame shift mutagen. So keeping that concept understanding in our mind, we will look at the observed results. So what we see here, the mutation caused by X is reversed by nitrous acid. It is also reversed by hydroxylamine, but it is never reversed by the acridine orange. It means the X definitely does not do the frame shift mutation, and the nitrous acid does a frame shift mutation, transition mutation. So the nitrous acid can reverse another transition. Okay, so as I was saying that the mutation generated by the X, if reversed by the nitrous acid, the nature of the X has to be that it generates transition type of mutation. So now the X generates the transition kind of mutation. Now look at the Y. Now if you look at Y, it generates a mutation, and that mutation is not reversed by nitrous acid. It is also not reversed by the hydroxylamine, and it's also not reversed by the acridine orange. It means it is not a transition type of mutation, because if it was a transition type of mutation, it should be reversed by the nitrous acid. It means it is not a transition type of mutation. Similarly, it is not reversed by the acridine orange, so it is also not a frame shift kind of mutation. So it is neither transition kind of mutation, neither frame shift kind of mutation. Then it should be transversion kind of mutation. Transversion kind of mutation. So that should be in our mind, that it should be the transversion kind of mutation. Now we look at the Z. Now we look at the Z. It is not reversed by the transition kind of mutagen. It is also not reversed by another transition kind of mutagen, but is reversed by the frame shift mutagen. So it means the Z is also a frame shift causing mutagen. So now once we have identified, or maybe we have dissected out that the X is a transition kind of mutant, Y is a transversion kind of mutant, and Z is a frame shift kind of mutagen. So now we look at the options, assuming that X and Y Z caused any of the three type of mutations: transition, transversion, or single base deletion. Single base deletion, definitely the deletion will create the frame shift mutations. So keep that one in mind. A single base deletion. If there is a two base deletion, that will also result in frame shift mutation. If there is a 3 nucleotide deletion, it will result in no frame shift mutation. Maybe single amino acid deletion, so that will not cause a frame shift mutation. So single nucleotide deletion and 2 nucleotide deletion or addition will definitely result into the frame shift mutation. The single base deletion means a frame shift mutation. So we know the Z causes deletion mutation. Z causes single base deletions, it means it's okay. Y causes transition, no. Y causes the transversion. X causes transversion, no. X causes transition, yes. Y causes transversion, yes. Z causes single base deletion, yes. Third, X causes transition, yes. Y causes single base deletion, no. Z causes transversion, no. X causes transversion, no. Y causes single base deletion, no. Z causes transition, no. So the answer will be 2. X causes transition, Y causes transversion, and Z causes single base deletion. So the option 2 is correct. So similar kind of question, basically this is the standard kind of question which is asked almost every year. I mean very frequently, not every year, very frequently in CSIR from the section of the DNA damage, mutation and repair. Basically this questions comes in the section of the inheritance biology, but the basic mechanism has to be explained in the fundamental process. So I brought these questions in my section of the fundamental process. Okay, we have discussed about all these things, and based on all the things we have discussed yesterday, so there are certain questions that will be like a recapitulation question, and we will discuss here that has been asked in different times in CSIR. For example, here, error-free repair or double strand. Error-free repair or double strand breaks in DNA is accompanied by. So this is a question not to discuss right now. We will discuss it later when the corresponding section will come. This question: which of the following mutagen is most likely to result in a single amino acid change in a gene product? So single amino acid change means, let's suppose there is a protein, and in the protein there is, let's suppose, amino acid G, and in the polypeptide now this G is converted by, let's suppose, lysine. The lysine is represented by K. So let's suppose it is replaced by glycine is replaced by lysine. So this will be a point mutation, and in the point mutation G is replaced by K. So maybe glycine is replaced by lysine. So this we call amino acid substitution reaction, substitution mutation, amino acid substitution mutation, and sometimes this is also called as point mutation. Okay, and as we discussed last time that the point mutations are generally due to transition and transversion kind of mutation, and the transition and transversion kind of mutations are generated by the methylation, the deamination, and then the oxidation and reduction processes. So here what we see, the example, what kind of mutagens are given. Acridine orange, we know that is the DNA intercalator. Now we see the ethyl methyl sulfonate, this is alkylating agent, and then we have a ethidium bromide. Ethidium bromide is also intercalating agent. So these two, acridine orange induce frame shift mutation. Ethidium bromide also induce frame shift mutation. And the X-ray induce DNA double strand break. Double strand breaks and single strand breaks. The single strand breaks will finally result into a double strand breaks, will result into DNA double strand breaks, and these single strand and double strand breaks will result into non-homologous end joining repair, and the non-homologous end joining repair results into deletion or addition of some nucleotides. Addition or deletion of some nucleotide, and that will result into again frame shift mutation. So what we see here, the acridine orange, X-ray and ethidium bromide, all of them will finally result into the frame shift mutation, while the ethyl methyl sulfonate will act as a like alkylating agent, and the alkylating agents results into base modification, and the base modification will result into transition or transversion mutation, and that will result into amino acid substitution mutation. So now we know that the answer will be ethyl methyl sulfonate. So this will result into a single amino acid change. Change means substitution. One amino acid will change into another amino acid in the polypeptide. The answer will be your third, ethyl methyl sulfonate. Another question is, that is the CSIR. All of these questions are CSIR questions. For your reference, there has been number given, like a CSIR December 2015, and the booklet code A, and the question number 30. So you will get all these things in our booklet, so you don't worry about that. So you will get everything, so and you can pause the video also. If you need to look at something more carefully, you can pause the video. So the question says, which one of the following chemical is a DNA intercalator? We already know the DNA intercalator are ethidium bromide, acridine orange, proflavin, etc. So here is the acridine orange. Okay, so that is the intercalator, we already know, so the answer is 3. Another question is June 2014, question number 51. Bromouracil is a base analog that can cause mutation. So we already know the bromouracil is the analog of T. It is the analog of T. So during the DNA replication, it can incorporate at the place of T. For example, there is a DNA, and this is the base pairing A and T, and during replication, rather than T, bromouracil can incorporate here at the place of T, and here will be T, and this will incorporate A. So that during DNA replication, if the bromouracil is present in the medium, and from the medium it goes inside the cell into the cytoplasm, and from the cytoplasm it goes into the nucleoplasm, and it is there in the nucleoplasm, and when the cell is replicating in DNA, during DNA replication, data DNA at the place of T, this bromouracil can incorporate. And once the bromouracil will incorporate, it will, in the next round of replication, the bromouracil will behave in a different fashion. It will behave like a T, and it can behave something differently also. So here the question is, the bromouracil is a base analog that can cause mutation when incorporated into DNA during the replication. Which of the following is the most likely change that bromouracil induce? As I said, the bromouracil generally it behaves like T, and though it is an analog of the T, but it does not base pair with T. Rather, it base pair with C. It base pair with C. And because of that, your AT base pairs convert into CG, or say your TA get into CG. Your TA base pair gets converted into CG. So that is the kind of transition mutation. It causes the transition mutation. Okay. So which of the most likely change that bromouracil induces? The bromouracil will induce probably this one, TA to CG transition. And if we go back in the list of the mutagens, if you remember the list of the mutagens here, okay, so in the I have not included the base analogs, but I have explained you this in detail. For example, this one, sorry. Okay, bromouracil. Bromouracil, okay, so it hybridizes with not C with the G. Okay. Okay. So the bromouracil, it will rather than T, yeah, this is the problem. Bromouracil will not, it should behave like T, so it does not behave like a T, and it does not hybridize with A. Rather, I missed something in my mind. Rather, this bromouracil will hybridize with G. And in this way, the TA, this one, so this one, the TA, generally it should behave like TA. So it will behave like a C, it will hybridize with G, and this will become something like, in the next round it will become like a CG. So it will be TA to CG kind of mutation. So it will be from TA to CG, like this one. Okay, this one. Okay, sorry. It was A, it was supposed to be T in a DNA, originally, but it becomes something like this AU during the DNA replication. So this was the original AT, it become AU, BRDU, and then this A gives AT in one replication. In another replication, the BRDU makes G, and next round of replication, the G will incorporate C, and the BRDU will incorporate G and BRDU. So here, after 2 rounds of replication, AT will become GC. AT will become GC. So the AT will become GC. AT will become GC. AT2. Which of the following is the most likely change that bromouracil induce? AT2 GC, AT2 GC, AT2 GC. So what will happen? AT2 GC, AT2 GC. Okay, yes. So this will be the first will be the answer. AT2 GC, AT2 GC, or TA2 CG. This is same TA2 CG. So TA2 CG, this will be the answer. The answer is the first. Okay, next what we see, some DNA errors occur during DNA replication that are not corrected by proofreading activity of DNA polymerase. They are corrected by a specialized repair pathway. Defecting the activities of some of the following enzymes and blah blah. We will not discuss it right now. We will discuss when we will discuss the repair systems. Next one is the December 2011. The base analog to aminopurine. Base pair with thymine and can occasionally pair with cytosine. The type of mutation induced by 2-aminopurine is transition. We have discussed this in detail in the last class. The 2-aminopurines and the BRDU, they all cause the transition kind of mutation. So the answer will be transition.